Namaste students, I am Jayoti Dr. Aziz Mohammed from Department of Food and Biotechnology, Faculty of Agriculture and Veteran Science in Jayoti Vidyapeeth University of Jaipur. Today I am going to discuss about the different methods of the microbial controls. In the previous video I discussed about the heat control of the microorganism. So it is the continuous part of the previous lecture, which is related with the other physical methods of the microbial control. What are the different methods which can be used for the control of the microorganisms using physical methods? So the first physical method is filtration. Microorganisms can be removed by the method of the filtration. We can use some filtration method for the removal of the microorganisms from any kind of liquid solution. Okay, we can also separate it from the gas. Okay, removal of the microbes by passage of the liquid or gas so a screen like material. It's small pores. We apply a membrane filter which have a particular size of any pore size, and it is used to sterilize heat sensitive materials like vaccines, enzymes, antibiotics, and some culture media. It is high efficiency techniques. So what are the different techniques which are used in the physical method of microbial control in the filtration? The first is the high efficiency particulate air filters, that is the HEPA filters, and they are used in the operating rooms, and they are used in the separation of bacteria from the air. And actually these HEPA filters are used in laminar airflow. They are using the laminar airflow because these HEPA filter will separate the outer environment from the inner environment of the laminar airflow, and they definitely clean the air. These HEPA filter are used to remove the microorganisms which are present in the air. Similarly, this is for the air, okay, but from a liquid solution we can use membrane filters. The membrane filters are of uniform pore size, and they are used in industry and research of different sizes. The HEPA filters are of the membrane filters are of 0.22 and 0.45 pore size, and they are used to filter most of the bacteria. Okay, and they are used to separate respiratory mycoplasma and viruses. Actually, these techniques are used in the downstream processing. These membrane filters are used in the downstream processing, and we use these techniques in the antibiotics, suppression of the antibiotics, suppression of the vitamins, suppression of microorganisms from the vitamins, vitamins, antibiotics, wine. You can also apply this technique for the wine also. Similarly, the pore size of the 0.01 micrometer pores are also used, which are used for the separation of viruses and some large proteins. Now, next technique is the low temperature technique. The low temperature technique is also sorry, it is also effective for the separation of the microorganisms organism from some specific products, and effect depends on the microorganism and treatment applied. The low temperature techniques include the refrigeration and the freezing. In the refrigeration, temperature from 0 to 7 degrees centigrade is used, and it is bacteriostatic, means it is not killing the microorganism, it is inhibit It is used to inhibit the growth of the microorganism. It is used to reduce the metabolic rate of the microorganism so they cannot reproduce. It is not used to kill the microorganism. It is only just to controlling of the microorganism, not to kill the microorganism. It is just to stop the growth of the microorganism. Okay. Now freezing. Freezing is also a low temperature technique, and in this technique, temperature below 0 degree centigrade is applied. The freezing may also have some techniques like flash freezing. In the flash freezing, microorganisms are not killed. Okay. In the slow freezing, it is slow freezing is more harmful than the flash freezing because some ice crystals are formed, and these ice crystals can disrupt the cell structure of the microorganism. About a third of the vegetative bacteria may survive 1 year, and more parasites are killed by a few days of freezing. So, slow freezing is more harmful for the microorganisms to be killed. Next physical technique is the radiation. So, radiation are the some light which are applied on the microorganism, like UV radiation, like ionizing radiation. So these radiation will cause damage to the microorganisms. So what are these radiations? So first one is the ionizing radiation. Some ionizing radiation like gamma rays, X-rays, electron beams, or high energy rays have short wavelength. Okay? They have a wavelength less than 1 nanometer, and this the disclosed electrons from the atoms form ions, and they cause mutation in the DNA and can produce peroxides in the microorganism. If these mutation and peroxides are produced in the microorganism, then they can be killed. And this technique is used to sterilize pharmaceutical and disposable medical supplies, food industry, industry using the ionizing radiation. Because it is a simple method, it is not it will not disturb the chemical nature of the food. So ionizing radiation are more used in the food industries. Disadvantage is that uh, it penetrates human tissues and may cause genetic mutation in human. Okay. So what are the different forms? 300, 400 wavelength, 500 wavelength, 600 wavelength, 700 wavelength, 750 wavelength. So these are the wavelength, and these wavelengths belong to the visible light, and the light below the visible light is known as the radiation, and which is very harmful for the microorganism. Next is the ultraviolet light or non-ionizing radiation. Uh, Wavelength is longer than 1 nanometer. Ultraviolet light is longer than 1 nanometer, and it damages DNA by producing thymine dimers. So there are some thymine dimers are found in the microorganism DNA, which cause them mutation. And it is used to disinfect operating rooms, nurseries, cafeterias. And the disadvantage of this technique is that it may sometimes damage the skin, eyes, and it doesn't penetrate paper, glass, and cloth. So they are not sterilized. So these all are the some different techniques by which we can sterile our different type of we can sterile different products from the microorganism using the physical methods. So in this lecture, I discussed about some physical methods of uh, killing microorganisms by filtration. Okay, filtration is also a very important method for the killing of the microorganism. Low temperature techniques were also discussed in this lecture, including the refrigeration and freezing. Radiation is also a technique which is a physical method of the microbial control. which include ionizing radiation and ultraviolet radiation. I hope the students have been better understood all these things. Uh, the question which can arise from this technique in this lecture is that explain different methods for the control of the microorganism using physical methods. What are the different physical methods which can be used to control the growth of the microorganism, including uh, mm, including low temperature, including filtration, and including radiation. Okay. So the examiner can also ask you about what are the different techniques which can be used to control the microorganisms using heat. Okay, what are the heat-based techniques for the killing of the microorganism? So I hope you can answer all these things. So my lecture is over here. If you have any query, you can ask me. So thank you very much.